information. So we've got our student lesson guide. So right over here, that's going to open a window, and that's going to pretty much walk you through. And there's another place for you guys to download the things, the materials that you're going to need. So let's go ahead and let's open this up, because I think this is going to be helpful. So here we have our table marker, okay? So I'm not gonna have this up the whole time, but really what the most important thing about this is specifically understanding that these are the markers for each allele repeat number. So remember that when we're talking about these tests, specifically what we're doing is doing a PCR test, which takes the segment of DNA and duplicates it, cuts it up and duplicates it a lot of times, so that way we can look at different DNA. And so when we're taking a look at the DNA, we really do want to focus on the base pairs, the allele, the repeat number, and how many times, right? This is the base pair length, the BP. And we're going to use this information to match up our victim. So the first thing is that we need to understand the background. So the background of the situation is that we're on a Midwest campus, right? Let's see here. So here's the story. We have these campus assaults. There's this college campus where 3 females were assaulted, all within 6 months of each other. And in the last attack, there was a blood stain on a knife, and then they were recovered by the police. And so they were looking at the DNA and analyzing the DNA from the blood stain on the knife, and they're trying to match it to either the victim or the suspect. Thank you. So we use this idea of the polymerase chain reaction along with fluorescent labeled markers to create this evidence, right? So we know that there are 13 STR markers in the United States are the ones currently used by the FBI's CODIS system. And then we've got this electrophoregram, shows the number of repeat units for each STR marker in the DNA sample, and this is specific to the individual. So we know that we have a base pair scale, there's a relative fluorescent unit scale, the number of copies, and an AML for the sex chromosome. So that's how we can determine whether or not they're male or female by the X and Y chromosomes. So here we have a picture, and so we can see that there is an X at 1,000. So if we take a look at the peak, so we're not necessarily looking at the branches here, right? We're taking a look at the peak, and at the peak there is about 1,000 X and there is 950 Y. At the peak, there are about 2,600 here and 2,700 for the D3S, right? So when we're trying to read through it, we're trying to understand how many base pairs there are. So these are the base pair numbers, okay? And we've got the relative fluorescent units, meaning how much it shows up under the fluorescence. And then we've got our STR loci, and this is the location of the gene, okay? So this is kind of what that gene would encode for. So if you have the numbers, so the upper number is the number of STRs and the lower is the RFU, which I don't remember off the top of my head. And in the beginning are the sex chromosomes. So this is how you can tell whether they have an X chromosome and or a Y chromosome. This is if they're heterozygous. So remember that heterozygous means that they have 2 of the same alleles. Okay, so that means that they have the exact same number or the same type. So that's either 2 dominant or 2 recessive. So that is what they mean by heterozygous for this gene. And this is homozygous, meaning they have the same. Okay, I messed up. Heterozygous is when they have 2 different. That makes more sense. Homozygous is when they have the same. So you're going to just see a lot of the same alleles repeating itself. So that would be like if you had big B for brown hair or little b for blue eyes. It could be either or. And then that's homozygous. And then this tells us the individual DNA profile. So this is what their count is going to be for each one. Okay, so questions on how to read this. Remember, I'm doing this with you too. So really what we need to look at is whether or not they're homozygous or heterozygous, so whether there's 2 or 1. And then we need to pay attention to the sex chromosomes, and we need to pay attention to these upper numbers, right? 12, 13, 18, 19, 20. That's the individual, and then we've got our individual DNA. So this would be the person's DNA. Okay, so then we've got our example. So we've got our example sample and then our suspect. So if we know what the suspect DNA is going to be, we can then use the locus and the heterozygous, the base pairs, to determine, compare our results. So once we're able to do that, we can then look at our lab and we can start identifying who the suspects are. So the suspect versus the victim, who does the blood belong to? So again, you're going to be given an electropherogram. You're going to analyze the alleles and then prepare a DNA profile and then determine whether each suspect can be included or excluded in the crime, and then search the text version database and determine if there are any matches. Summarize your conclusion. So what that means is, here we go, we have it again. So we want to download the laboratory instructions if you haven't already. And so when we look at it, it's going to look like this. Now let me go ahead and enable editing, and let's zoom to 200. And I'm going to have this be side by side, specifically because we really do want the information. Let's see. Okay. So if you haven't already, go ahead and open this document up. I want everyone to be doing this with me. I'm going to, at the very least, get you started and then give you some time to work on this on your own. But like we said, we know that the story goes that on a Midwest college campus, 3 females were assaulted within 6 months, and they found a bloodstained knife. When they were questioned, they said the attacker was a tall man who wore gloves and a mask, and he threatened them with a knife and took them off a main campus quad into a wooded area where he beat them. But even though they didn't see their face, they were able to scratch him. And remember Lockhardt's exchange, right? So they were able to get some of the blood samples and scrapings from underneath their fingernails and their bloodstained clothes. So in the course of the investigation, police detained 4 possible suspects. All the suspects are white males. Although suspects A, B, and C could not be held because the victim could not identify them from a lineup, the police did manage to get their saliva samples. So unknown to the suspects, police collected their discarded cigarette butts and drinking glasses so the saliva could be used for DNA testing. And we've got our suspect D was arrested shortly after the last assault. He had scratch marks on his arms, and police confiscated a bloody pair of jeans from his apartment. They took that as a DNA sample from him and the clothing. The evidence has been sent to you, the forensic scientists, and now you have to do the lab. So the forensic scientists ran the PCR with the STRs for all the evidence samples, and you have collected these 4 specific samples. Okay, so these are the 4 specific markers that we're going to be using, and they've separated the PCR into DNA fragments by capillary electrophoresis, and have obtained the electro in the sample. So we've got these different samples, and what we want to do now is take a look. So for each STR marker, locate the base pair length. For the peak or peaks, and then for example, for victim 1, there is 1 peak for the STR marker. The peak is located at BP 272. So what we're going to do is, if we move on, we can even use they have the DNA and they have the information right here. what we're talking about in this, let's move this one to the right, and then we can look at this. Okay, so they're telling us, they're giving us an example on how to do it first. So victim 1, there is one peak for the STR marker D18S51. That's this guy over here. They said that the peak is located at BP at base pair 272. So we look at this table, the peak. So remember, we're not looking between the two lines, but the peak right there. Okay, Caroline, just give me one more moment. I'm just going to get some started, and then I can help you with the lab that you have questions on. And then, so we look at D272. Now, there's only one peak. So what does that mean for this victim in terms of heterozygous versus homozygous? Have two different alleles or two of the same allele if they have one peak?
I'm sorry, I'm confused. What is the DS, the light blue one?
This guy?
Yeah.
It's just a marker. So we just look at specific parts of the DNA, because remember that the DNA is this huge thing. So we really just focus on 4 markers, and which just means these 4 specific placements in the genome, and they can use that location, that gene, and they can use that DNA sequence to determine what the alleles are, what those genes are. So I don't know exactly, I can't tell you exactly what these are all for, but this is a specific gene sequence that they are looking at in this individual's blood that came up. Does that make more sense, Courtney?
Kinda, yeah.
Okay. So I know it kind of looks a little confusing, but really what you need to know is that when they were looking through the blood, this is the couple of instances that they're able to be like, okay, this is clear enough that I can use this to compare the DNA from blood from other people. And then we're going to look at all the same genetic markers. We're going to look at all the same genes so that we can compare it. So that's just what they're saying. You can use different markers. You can use different genes, but this the one way that this forensics scientist looked into the information. So, does that make sense so far? Okay. So then, right, we notice that there's one peak for the STR marker, right? And then we know that the peak is at 272. It's only one peak, meaning they have this 2 of the same allele. So then we're going to use this, right? So it says now use the CODIS STR marker table. So you're going to click on the table. In this case, it's in the bottom right-hand corner. And then you can use that table to convert the base pair number to the allele repeat number. So we know that it's at 272. So if we take a look at this, and we've got this is the gene marker that we're looking at, and the base pair, well, then we see that it's at 272, so we know that they have 2 9s location. So then, right, so here's the table. So we take a look, this, the allele repeat numbers, right? So we know for this one, 99, and that's, or sorry, this is confusing. Yeah. This is the right one. Okay. 272, if we take a look at the allele number, it's this one. So now we know that victim 1 is homozygous for this marker, specifically because there's one peak, and that means that they have 2 of those alleles that have the number 9 from both parents, and then you can record that as 99 in the DNA profile. So when you're trying to create the DNA profile for your victims, that's where you're going to write it down. So now if we take a look at victim 2, let's look at the AML, right? So we have at this AML, we know that this is going to give us our sex, right? So whether it is male or female, and we need to look at the base pair. We see there's only one peak, which means they're going to have 2 of the same. So if we see this, and this is 100, 110, so each of these tick marks go by 2, we see that this is 102, 4, 6. So 106, if we use the table for the animal, we scroll down, 106 is an X. There's only one peak, meaning they have multiple X. So that means they have two X's. So that means our victim is going to have XX in this site, in this spot. And then that's what you're going to do for the DS3 and for the rest of the markers for each one of these. Okay. Now, like we said, you're going to go through these. So if you go through the online version, you'll have the victim. So here's the DNA for the victims. And then on the next slide, they should have the DNA for the shirt. Okay, so then you've got the DNA, and that's what you're going to do to fill out this table. Okay, so once you've filled out the table, where did I go? Where did my big, here it is. Once you fill out this table, then you can start to draw conclusions. This is all the DNA from the different suspects. And then when you look at suspect A, B, C, and D, you can start to compare which one you think it is and then answer the conclusion questions. And then you're going to need to do part 3. Okay. So that was like a really quick run through. I want to pause here just so that way I can answer Caroline's question. But is there any questions about this lab? Ethan, Courtney, did you guys have any, does this make more sense now? And I can always meet with you during a help session if you want to spend more specific time on each one of them.
We're just matching. Yeah.
Mm-hmm. Yep. And then we're using that table to determine this allele number. And then that's what you're going to put over here, right? So like, for example, they had one peak and then they looked at this table. So if they had 15, then that must mean that, where's that other guy? That must mean if they had 15 and they're talking about the VWA, it means that they had one peak at 172 base pair, right? And that's how they know it's 15, 15. That makes sense? And then you're comparing it and trying to determine which suspect was whose blood is present. Good? For now, we can always meet again. This does not have to be the one-all be-all for it. But at least we can get started on this. Okay. Caroline, which one would you like to go over?
I just want to say I'm sorry. I didn't think about the timing of my comment. So I'm sorry for that. I don't actually have a question. I was just like, somehow in my brain, I was like, send that. No, I'm sorry.
No, that's totally fine. You know, I just want to make sure that I'm helping you guys, right? So like if that means, if you've done the DNA lab, did you do the fingerprint one yet? They just muted you.
I haven't done that one yet. I'm getting to it.
Yeah, so let's go over that because everyone's going to have to get to that, right? So even if the DNA one is for today, we can still go over the fingerprint one. So again, remember that the fingerprint, you're going to learn about it before you get to it. And the fingerprint lab is not technically until later on this week, but we have 3 labs this week. So it's a lot. And so remember that this one is really, the first one was about matching the DNA, and it's going to be similar for the fingerprint one too. So I'm not going to go too in depth to it just because if some of you guys haven't gotten to it yet, it's going to make way more sense once you do once you do less than 310 and then you go into the laboratory. But we can at the very least start to look at what needs to be done. So when we go to the fingerprints, right, we're going to go through the introduction. So let's just go through this together just so we can all be on the same page. But I will tell you, when I was looking through this, I learned so much about fingerprints. But it's really fascinating because you could even look at your own fingers and start to determine the characteristics, right? So in this investigation, what we're looking at is a pretty high heist crime. Someone came in and stole a highly praised painting, but they left some fingerprints behind. So what we're going to do is look at the fingerprints from the crime scene and investigate it and try to determine to whom they belong to. So again, we're going to download this information. We have this evidence pack, and then we've got the comparison tool, and we've got the lab instruction and the text version. So all of that is really just documents of what they provide for you here and in the lab. So an expensive painting was stolen. You're going to have to answer some of these questions when you work through the online activities. So as you click through, you're also going to notice that their instructions are provided to you online if that's where you choose to do it. Or you can download the documents and print them out. It's really up to you. But like we know, there was an activity, there was an expensive painting that was stolen. All of this, of course, is fictional. And we have this fingerprint examiner. So we've got these printed, right? So you have to examine the fingerprints recovered, and you're going to be given a 10-print card similar to the one here to show a prior conviction, and this is a permanent set of individual. So the main thing that you need to know when you're going into this lab is the 3 different types of patterns. We've got loops, we've got whorls, and we've got arches. So this is a loop, right? So this is, what we would think of. Like if I look at my thumb, I realize I actually don't have one on my thumb. However, I do have one on my middle finger. I have them on a couple of my middle fingers. It's actually really fascinating to be looking at my fingerprints, knowing these things. But a loop is going to be is a ridge beginning and ending in the same side of the print. So if you take a look at your own fingers, you can see whether or not you have one. But really, it's going to start at one end and come back. And that's what's considered a loop. Then we've got something called a whorl. A whorl is the circular or oval spiral found in the center of a print. So that's when you're going to take a look at the very middle of it. And if it kind of looks like a kind of a circular pattern, then it's considered a whorl. A whorl. And then we've got arches. So arches are what you would think of as an arch. They don't necessarily start, they start from one end, they go to the other. They don't start from the middle, they actually go from across. Okay, and that's going to be the fingerprint on, those are the 3 patterns that you're going to want to identify. They're not always easy. So when we talk about deltas, one way that we need to think about it, right? So when we talk about the variations of the 3 basic types of fingerprints, sometimes they don't all have it. So we need to distinguish the whorls from loops and arches. And if you're having a trouble classifying, then you're going to want to look for deltas. And they're going to explain what deltas are, but I'm assuming that deltas have, some sort of structure to it. So if a whorl has 2 deltas, and we think of a whorl as the one that's coming from the start at the end, then we might think of them as the beginnings, right? The end point. So look at the fingerprints and identify the finger type activity. How would you classify each one? So if we think about it, you may not be able to see all the deltas because the print doesn't have enough area of the finger. And then identify the fingerprint. So those are some practices that they've included for you. So taking a look at this, what would you say this is? Is this a whorl? Is this a loop? Or is this an arch?
I think it's a whorl.
It's a whorl. Good. What about this? Is this an arch or a loop?
Arch.
Good. And is this a whorl, a loop, or an arch?
Looks like an arch.
Okay. What else do we have? Ethan, any ideas? The answer is a combination of a loop and a whirl. It has one delta. So I guess these are the deltas you're looking for. So if it has one delta, then it's classified as a loop. So yes, I could see what you're saying, Caroline, how it has that like arc pattern over here, but then it turns into a loop. And so we look for these like moments of crossing. So thinking about that, okay? And then let's go back and see what they say for their answers. So this fingerprint has two deltas. It's clearly a whirl and has two deltas. So yeah, those points where they cross, right? And they meet. So those are considered deltas. Those are those points. And then we talked about how this one was a combination. And then we've got this whirl. Well, I thought it was just, I would have figured that this would have just been an arc. But it looks like it's a little bit of a whirl and they have one delta, so it's considered a loop. This has one delta, so it's also considered a loop. And this one, what is this one gonna be? See if you can look.
Weird.
I know, it's a little difficult, but let's see if you can locate how many deltas there are. How many do you see?
I see 2.
Okay, so Ethan sees 1, Caroline sees 2. Courtney, how many do you see?
I'm looking and I only see 1.
You only see 1? All right, let's find out. There is 2. So there was this one delta that was very, very clear to us, right? But then there was this area that was a little bit less, but there, right? So then if it has 2 deltas, it's classified as a whirl. This is going to be a fun lab.
That's really cool.
Yeah, that's what I'm saying. Now I'm like looking at my fingers. I'm like, what do I have? I wish I had I want to know what I have. Anyways, that can be done on my own time. Anywho, so now we're starting to see, and so I would really I would really practice that before you start the lab, because then we're going to get into this equation, right? So here's the thing. So when we are trying to do this, we have this kind of fraction that we're going to look at the pair, and each one, so depending on where you're starting, is going to have a different value. So if you're talking about your thumb, your, let's see, your right hand, so your right thumb and your right forefinger, if you have it, it's going to be 16. If you don't, it's going to be 0. So so you'll be asked to make a Henry classification of Mr. Kurtz's fingerprint record. Recall that Henry classification assigns a number for a whirl and is 0 for all other types. In the example below, the right index, the right middle, the right little, the left thumb, and the left ring fingers have whirls. So it's either you got it or you don't. You either have a whirl or you don't. And then depending on which pair is going to give you a specific number of value. So for example, if they said that in the example below, the right index, so that's this one, the right middle, and oh, that's right over here, and the right little have whirls. And so they're going to be assigned in a number, right? So for example, the right index has it, the right thumb does not, right? So right index has it, so they have 16. The thumb does not, so then they have 0. They said that the right middle has it, so that means this guy's going to have it, and this guy does not. So then we have an 8 and a 0. Both the left thumb and the right little have it, so they both get this number of 4 and 4. Neither the left middle nor the left index have it, so they have 0. And then the left little has it, but the left ring does not, so this value gets 1. And then we have this number of 21 over 14. Okay, and then we can use this number to compare it to our victims or to our people, right? So then you're going to have an opportunity to practice. So if you take a look, the right index and the right thumb, And then you'll be able to work through this and use your guides. Okay, so then you'll be able to use the guides here, right? Remember that they tell you the Henry classification. If this is really confusing right now, that makes sense, right? Because remember, you still have to go through the lesson for fingerprints. So then you'll have a better understanding of how the Henry classification came about. It'll give you more specific examples about how you can summarize and identify the different patterns. And then you're going to make a concept map. You're going to review and compare. And then that's what you're going to do for the fingerprint lab.
Thank you.
Yeah. Does that make sense? Or did I just I think it'll make even more sense once you go through the lesson. But so far, how are you guys doing with this?
It makes sense. Just matching the patterns of the fingerprints to the patterns of the fingerprints found at the crime scene, and figuring out who did it based on that. And pretty straightforward.
Yeah. Yeah, and I think the hardest part is really just going to be when it comes to identifying the patterns, the understanding the deltas. So really looking for those deltas, and then if it's confusing, and then recognizing the pattern, and then giving the pattern the numerical value. So like, for example, if you took a look, hold on, let me get back to it. If you take a look, nope, that's not the one. Here we go. If you take a look here, right, we know that there is, it goes 16, 8, 4, 2, 1. So just kind of knowing that it goes by a half down the line, I think is going to be it's just going to be difficult about knowing, okay, this is a 16 or a 0. This one is an 8 or a 0. This one is a 4 or a 0, right? So kind of keep that in mind when you're going through this lab, to remember the numerical value that you're going to give it. And it really only gives it a value if it's a whirl. Okay, and then you can use this number to compare to the others. Okay.
Do you think they have a machine that searches for whirls?
I do. I think now they do, yeah. I think now they have machines that do this analysis for you. But it's interesting. I don't seem to, I wonder if there was, I wish there was a way to there we go, zoom in. So let's do this first one together. So this is the first right thumb. So what do you guys see? What pattern do you think this is? Or I guess really we just need to know, is it a whirl or is it not? Do you guys think this is a whirl?
Honestly, on my screen, it's just too blurry to tell.
Okay, that's fair. Well, what about this one?
The first one looks like it would be an arch, like the center. It looks like it's smudged, so it's hard to.
Yeah, it is. It's kind of hard to tell. So, I mean, then what are we going to put for it? Like, should we put just a 16 or a 0?
I think a 0.
A 0, right? That's what I would put down too. And then we've got our right index. Does this look like a whirl? We've got one delta right there. And do we see another delta? There's one delta right there. And I think I see one over here. So I'm going to put 16 because I think that the right thumb is a whirl. Why isn't it letting me put? That's frustrating. It won't let me put the 6. Pair 1 fingerprints are assigned 16 if they are a whirl and 0 if they are anything else. I know, that's why I was putting a 6 in there. Okay, so then if I put, so it was 0 on the bottom. Oh, because I read this wrong. Okay. That makes more sense. I'm really struggling, you guys. There we go. But the number is in the wrong place. Okay, now let's look at the ring finger and the right middle finger. So we've got our right middle finger and our right ring finger. So what is this, a whirl or no?
It looks like it.
It does. And we've got one delta, and I see we've got like another delta over here. So I'm going to say it is, and that is the right middle.
So the delta.
It muted you.
Thanks.
What's that?
The deltas kind of make like triangles in the fingerprint where they meet. Is that what I'm like seeing? They kind of make triangle figure thingies.
Yes. When they mix, right? When they start to mix, that's when we start to see that there's like this one spot. Like this one has a pretty clear delta. So when the patterns meet, they kind of create that. Remember the delta is a Greek letter that we use. It's a triangle. That's why they're calling it the delta. And so there's this one area where they all kind of meet. It's also called a delta geographically. If you talk about them, like, you know, the different deltas, it's when things come together and meet and conjoin. So that's what we're also thinking right here. So there's a delta right here, and it looks like there's even a delta. Like there's a delta right here, and then it looks like there may even be a delta somewhere else, like over here. You can kind of start to see that pattern. So let's go on to the next. See what they show. So remember, there needs to be two deltas in order for it to be considered a whirl. So we think, do we think middle finger is a delta or has is a 0 or an 8?
Looks like an 8 to me.
Oh,
can you zoom back in?
Oh, yeah, of course.
Looks like a whirl.
Okay. Anybody else? Okay, so then we're going to put 8 for the middle. Now let's look at the right ring finger. Do we think this is a whirl or do we think this is anything else? Because there's like these arches and there's a whirl, right? Do we see more than one delta?
I think there's 2 deltas.
You do think there's 2 deltas? Okay. Ethan, Caroline, what do you guys think?
I really can't tell, but it looks like there's 2 joining places.
Okay. All right. Okay, so then let's go ahead and see if it's also a 0, right? Yeah, I understand, Ethan. So, you know, this is just practice. So, of course, you know, you're able to do it on your own computer, but at least we can practice. And you guys did a great job. You were right. They both had it. Yay. Okay, let's do one more. Okay, so for your right little finger, is this a whirl or is this anything else?
I don't think that one is a whirl. The little.
Okay.
I think that's an arch. I can see an arch.
You can
And
see
I
the
don't see
arch.
any deltas. Do you see any deltas?
Okay, so then let's go ahead and put in for the right little a 0 because it's not a whirl. All right, now let's look at the, here, this is, no, yeah, here we go. And there's the left thumb. So what do we think this is?
I think I know I see 1 delta.
Uh-huh.
But I can't really see another one because it's kind of like smudged.
Yeah.
Could you zoom in just a little bit? Because I can see.
I don't know if I can. You might be able to zoom in on your side. You have that like plus and minus.
Yes, I am zooming in. It is somehow getting further away. How is that?
Okay.
It zooms in on your faces. Anyway, that's annoying. It looks like there's a whirl. There's a whirl in the center, and I see a delta, like off to the bottom right, and a left middle. There's another delta.
Okay. So then let's put in a 4. Good. Let's check our answer. Right. You guys are amazing at this. Great. Okay, so do you want me to continue or do you want me to stop here? I can stop here and let you kind of work through it and then do this when you're doing your lab.
Yeah, I'd like to stop
Yeah.
and do it because I think it's really interesting.
You want to keep going?
No, I want to do it by myself, see if I can figure it out.
Great.
Yeah.
Okay. So do you want to do that? You want me to give you a chance to figure it out on your own, or you want to like do this lab separately? I just want to make sure I'm not like ending class early in case you guys wanted to give it a try and check. But I think what you're going to do it on your own time and do it, right?
Yeah, I think it would be helpful to like really read the DNA, like the
lesson and everything. That's okay. Good. Yeah. So okay.
Thank you for going through it.
Oh yeah, no problem. Like I said, I'm learning with you guys. So anyway, so those are like the things that I was going to go through with you guys. So if you have a better understanding with the labs, then you don't have to stick around. But if you want to get started on doing one of them together, I have about 10 minutes left. We can spend the 10 minutes getting started. So, Caroline, if you want to go on ahead and go ahead and do the lesson for fingerprints, go for it. And then, if Ethan or Courtney, you guys want to stay behind, we can go back to that DNA lab and work on that together. It's up to you.
Thank you so much.
You're very welcome. You're welcome.
I'll see you at lunchtime.
Yeah.